Hello everyone, today I will introduce the protein design tool ProteinMPNN in WeMol. The ProteinMPNN algorithm was developed by Nobel laureate David Baker and is a deep learning-based method for protein sequence design. This tool enables protein sequence design or restoration based on backbone structures and integrates with RF diffusion and AlphaFold 2 for applications in protein or antibody design. Finally, it provides the designed amino acid sequences. We can directly enter the visualization plugin WeView to operate. First, import the PDB file that needs to be designed. Check the structure. It is a protein-protein complex structure. There are missing residues in the structure, as well as sodium ions and water molecules. You can first use structure preparation to process the structure. Complete the missing residues in the structure and remove the sodium ions and water molecules. The structure processing is completed soon. Check the processed structure and delete the original one. Meanwhile, we can go to the File page. Save the processed PD structure to the personal data center or locally for easy viewing later. Then, we can directly use ProteinMPNN in design for protein sequence design. Enter the parameter input box. In chain, enter the chain to be designed. We choose chain of this time. Set the number of sequences to the number of sequences to be output, usually the default. Position type is the mode for designing residues. Among them, fix can specify that the residues in the next position remain unchanged during design. Design can specify that the residues in the next position can be designed while the other unspecified residues remain unchanged. In position, enter the specified residues. In this example, we take a segment of the sequence at the interaction interface between two proteins for design. The interface function in Analyze can directly analyze the binding sites between proteins. After the output results, select Interchain to view. It is found that the residues 118 to 127 at the C-terminal of chain are located at the binding interface. So we choose this segment of the residue sequence for design. After selecting the residues, click Sync Selection. The residue numbers will be directly displayed in the position input box. Note that the residue numbers here start from 1, not the original PD numbers. Sampling temp can set the residue sampling temperature. Omit Oz can specify the amino acid types that are not allowed to appear in the generated result sequences. Other parameter refers to the reference mode during design. Homomer is mainly used for the design of homomeric polymers. Soluble model can be used for the design of soluble proteins. After the calculation is completed, it will automatically jump to the sequence editor WESIC to view the design sequences. You can see that a total of 5 sequences are output. First, let's look at the chain names. T indicates the sampling temperature. Sample equals 1 is the design sequence 1. Score is the probability score of the designed residues. Global score is the probability score of all residues in the sequence. Seq recovery is the sequence recovery rate, the higher the better, indicating that it is more similar to the original sequence. To analyze specific mutation sites, the original PDB sequence was imported into WeSeq for comparison. The wild type of chain sequence was adjusted to the first row, and sequence alignment was performed. Conserved residues were marked with asterisks, and sequence numbering was converted to UID numbers corresponding to the original PDB sequence. The design sequence residues 1181-7 revealed mutations at positions 119, 120, 123, 125, and 127. These sites were visualized in detail using the Site View feature in WeView to assess their structural context and interactions. Subsequently, AlphaFold 2 was employed to model the design sequence. Select the first sequence in the output, press Ctrl to copy it, and paste it into the group with the B chain sequence. Adjust the order of the design chain, select these two chains and copy them. Return to the module interface, search and enter the AlphaFold 2 module. Paste the copied sequence into the sequence input panel of AlphaFold 2. Select the multimer mode and run the calculation. Once the structure prediction is completed, the results can be viewed. The predicted complex structure achieved scores of approximately 0.87, indicating high reliability, with the highest scoring model designated as ranked 0 PDB. This workflow demonstrates the integration of sequence design and structural modeling to validate the rationality and functional potential of the design mutations. The complex prediction results include a PAE heat map for the optimal model, which serves as an indicator of AlphaFold to ease confidence in the relative positioning of residues within the predicted structure. Dark blue squares in the heat map denote high confidence predictions, while light-colored squares indicate regions of lower confidence. The predicted complex structure exhibits high reliability, as evidenced by the heat map analysis. To further validate the structural predictions, the wild-type and mutant structures were superimposed in WeView. The RMSD value of 0.82 between the two structures confirms the reasonableness of the predicted model. Additionally, specific residues at positions 119, 121, and 125 were analyzed to assess the impact of mutations. The results demonstrate enhanced interactions at these sites following mutagenesis, indicating that the design outcomes are rational and functionally improved compared to the wild-type structure. This integrated analysis underscores the reliability and utility of the ProteinMPNN tool in protein sequence design and structural validation. This concludes the content of this video. Welcome to Try WeMol. Please give a thumbs up, follow, and share.